Good morning, everyone. First of all, I'd like to thank Celestina for inviting Julianne and myself to speak at the Inland Northwest Research Symposium. This is really a big honor for us, and we are very excited to share some of our research with you. And we look forward to any questions. I would like to, at this point, give you my email as well. It is KMURPHY2, so KMurphy2 at WSEDU. So feel free to email me anytime, and if you have any questions or would like to talk about this or other research, that would be fantastic. So the talk of my the title of my talk today is Soil to Society, and really want to focus on the intersection between the colleges of agriculture and human health at Washington State University. So I'm an associate professor in the Department of Crop and Soil Sciences. And my lab is the Sustainable Seed Systems Lab. And so here's a picture showing some of the crops we work on. And for those of you who live or have spent much time here in the Palouse, you know that we're really a wheat-based farming system here with about 2 million acres of wheat. So these crops are all really there to provide potential rotational crops for wheat. So we want to diversify the farming systems as much as we can. Farmers are very supportive of this. And so we focus on many of these crops: barley, quinoa, perennial grains, and spelt. We actually have breeding programs for each of these and have released several varieties as well. And then others like amaranth, buckwheat, hemp, or proso millet, we've done some agronomic research on, cropping systems research on, or nutritional research on. So these are our bread and butter crops, so for our lab. And I'd like to talk about one of the biggest challenges in agriculture, and in particular our field of plant breeding. And it's that food crops have decreased in nutritional value over the past 60 to 100 years. And this has been shown to be true due to a combination of factors. So it can either be genetics. So we've, over time, released less nutritious varieties. And it could be the environment. It could be soil health. There are many different factors. So we spend a lot of time trying to figure out what these factors are, how they contribute to nutritional value in these crops, and how we can improve them. Here, there are a few references here to show some of this, but there's quite a few more as well if you are interested in digging into this topic. And this isn't true for all crops, all locations, but by and large, this is something that we should be concerned about, and something that we do have the capacity to improve. And we can improve that through plant breeding. That's the primary purpose of our lab, but also we do some agronomy. Essentially, how do we grow crops, cropping system management, and we work closely with food scientists, soil scientists, all to do our best to improve the nutritional value, and I'll include heart healthy components of our food crops as well. So there's several papers in the literature suggesting that the intersection of agriculture and human health is essentially non-existent in many universities. And for example, universities don't necessarily have a strong college of agriculture and a strong college of medicine like WSU does. But even if they do, there's very little interaction there. And so the focus of one of these papers just shows that there's really a big canyon between these two colleges. So this is really the basis of what we're trying to do with our Soil to Society research. And this research was initially funded almost 4 years ago by the WSU Grand Challenges, or Strategic Reallocation Funds. And they provided $2.5 million, and we've been able to do pilot studies with those, hire new faculty with those, and put together a robust PhD and undergrad master's student program to really address some of these issues. And we're actively writing grants and getting some as well to continue these topics. And so what we're trying to do here is build a bridge between the College of Agriculture and the College of Medicine. And we realize that we speak different languages. There is a lot to learn, but it's incredibly fun and exciting, and there's great faculty on both sides. And we are now finding ourselves, if you see these two bridges here, you can just kind of imagine people walking along the bridges, getting closer and closer and intermingling. And really, well, I'll get to the point, but some of the best people to do this are graduate students. And so here are a couple of graduate students, Hallie Choi and Evan Crane. They are in this picture just to represent some of the work that we can do in the College of Agriculture. It's considerable, and so what we're trying to do is, do breeding, do agronomy, do soil science, and come up with the best varieties out of these, or the best methods of growing food, and then we can take these to either the WSU Bread Lab or the School of Food Science. That's Ju-Ming Tang there, who has ready-to-eat meals, which he is working quite diligently to improve the nutritional value of. That's Girish Ganjal below him, who's doing similar work, but a lot of really important work with breeders to help us identify different phytochemicals, et cetera, in our breeding lines that can then become new varieties. And so building bridges, as I mentioned, is really important for graduate students to be involved. So this is Julianne Kellogg, who will be speaking after me and talking about some of her research. Below Julianne is Ellen Mahoro, another graduate student in my lab there. And these are just examples. There's several more as well. And then linking with some faculty, new and administrators as well in the College of Medicine. So here's Glenn Duncan, Frank Carbonero, who's below Glenn, who is a new faculty hire through these grand challenges. And then on the other side, John Roll, Martine Peregri, Maynor Espidi, and then Pablo Monsevis. And there's others too, but this is just kind of a representation of the pipeline that we're working through to develop more healthy and nutritious foods. Okay, so I'd love to give you 3 examples of work that we do in our lab that focus on breeding and agronomy for health and nutrition. And these are very short examples just to give you an idea. And if you're interested in any of these, again, please feel free to email me and I'll be happy to talk more with you about them. So we'll start with quinoa. And many of you have probably eaten quinoa or certainly heard about it recently in the popular press, in the media, maybe in some scientific articles. And there's really been a large increase in interest and consumption of quinoa in the EU and in the US and many other places as well. And it's always touted as having a high nutritional value. And part of that, which you see often, is that it has a complete protein and therefore is considered a superfood. And so here are just a few articles that talk about this, but this is maybe 5 out of several hundred. But what these have in common with quite a few quinoa papers that have been peer reviewed, as well as they all say that quinoa is a complete protein. But if you look back, a lot of that is based on maybe 2 studies that looked at a couple of varieties in 1 or 2 locations, so very, very small sample size. So that led us to ask the question, are all quinoa varieties a source of complete protein? And by extension, which I'll get to in a minute, but are all locations that can grow quinoa, do they provide a source of complete protein? So, but we'll talk about the quinoa varieties first. Again, this is Evan Crane's work now. And So here's the question. So are all quinoa varieties a source of complete protein? And instead of just looking at 2 or 3, we started with looking at 100 varieties grown in Washington State, 4 different locations, and we sent these all out for wet chemistry analysis. So it could be analyzed for protein and each of these amino acids, among other traits. And if you look at this table, on the left-hand side, you'll see age and different categories of age. And then each column, you'll see different amino acids. And the main column I want you to look at is the one in bold, or the main row down at the bottom. It says all met. And so keep an eye on that one. But the ones in particular with the age groups, those show that the number of samples that do not meet the requirements needed for daily requirements for amino acids. So first of all samples met these requirements for all age groups, and all age groups is important here. But you'll notice some didn't. So tryptophan, for example, 94 of these samples met the minimum requirements for all age groups, but if you look at the under 2 age groups, you'll see that 6 varieties did not. So that's interesting to us. Lysine, even more so. So here, only 52 out of the 100 quinoa varieties met the minimum requirements. But again, all of these that didn't meet them were under 2 years old. So if you're over 2, so far so good. But then we get to leucine, and here only 9 varieties met all of these. Well, here leucine, the minimum requirements for leucine, and then if you look up, even though most of those were under the age of 2, there were some that were in the older children and adult groups too. So here, if you're an adult, still 8 did not meet the minimum requirement. So this is the first research of its kind to show these, especially with leucine, these limiting amino acids. So this is good news and bad news. So the bad news is quinoa isn't automatically a complete protein, which would be fantastic, a great source of plant protein for global food and nutritional security. But the good news is that quinoa is very close to being a complete protein, and some varieties are across all age groups. Most varieties are across the adult age group. And so since we are so close, if we focus our research efforts on improving leucine, lysine, and tryptophan, we can really solidify the complete protein status of quinoa. So current research, so that research I just showed is in review right now. We are continuing this research by upping the number of varieties. We're looking at 1,000 different quinoa varieties that are grown in 4 different countries, and then 350 quinoa varieties that are grown in several more countries. And these are across 5 continents, so really a global effort. We have great partners growing these with us, and then what they do is they send us all the seed, and we phenotype them for traits of nutritional interest, and in particular protein and amino acids. And so our goal here is to identify varieties as well as genes and genetic markers for protein and amino acid content. And so that's our goal moving forward. All of these 1,000 varieties have been sequenced. So we are now providing the phenotypic data, and once we have the phenotypic and genotypic data, we can really move forward in identifying the genes and genetic markers useful for selection of amino acid content. Okay, so that was quinoa. Now we'll move on to perennial grains. And perennial grains is a really interesting topic. We've been working on this for 25 years at WSU. It's a long-term project. You're essentially changing the life cycle of annual wheat to perennial wheat, which is a big deal. And but if you're interested in this in general, I would really highly recommend that you look up the Land Institute. They're a group in Kansas that have been working on this for 30 or more years. And they do this on many different crops, and they really have some phenomenal research going on there as well. Okay, so perennial grains are essentially, in this case, we're referring to wheat and wheat relatives, and the wheat, as you know, as you live in a wheat country, is annual. So after harvest, the plant is dead. But what we're trying to do is work towards getting the plant after harvest to regrow new tillers or shoots, if you will, from the crown of the plant. So in between the roots and the shoots is the crown, and then be perennial. And one of the reasons we're doing this is because the Palouse is very hilly, and there's a lot of soil erosion going on. So the less we have to till, plow, the more soil we can save for future use. And this picture is showing a perennial wheat breeding field, but I want to talk about how we get there. So we start with an annual variety. This one is Madsen, which has been grown in Washington for a long time. It's a good solid variety, but it's an annual, so when you harvest it, it dies. And then we cross it, and we cross it using traditional plant breeding methods such as hybridization with a pollination bag, with scissors, with tweezers. Very simple, although the success rate is very low in this, but it doesn't take much. So we cross it with this plant, which is a Thinopyrum species, so a wild wheat species. And the difference is here, this one is perennial, so it will regrow after harvest. But the harvest is very low. Yields are extremely low. So we do want to improve the yields and come up with a plant that is also perennial. And so that's what we're working on now. It is effort in progress, and a long-term research effort for sure. But we do have several very promising lines that we're looking at now. But I want to show you in particular a couple studies that talk about the roots. So the first one is a hydroponic root study, so just grown in water in a greenhouse. And the roots on the left are from a variety called Chinese Spring, a variety of wheat. The roots on the right are the same variety of Chinese Spring, but with just one small portion of one chromosome from the wild wheat species added to it. And so that, so it's a very, very similar variety. Wheat has 42 chromosomes, and so just a part of the 4E from the wild species has been translocated to the 4D genome and chromosome. So you can see the difference that makes in root growth. So we've been able to really hone in on where this difference is happening, how to find genetic markers for this, and really the impact it can have. So that's really exciting. But then, you know, that's in the greenhouse. We need to take that to the field. And so this is a work done by a recent graduate, Ian Clark. And what he did was take our perennial grain field trials and put in 2-meter long tubes, clear plastic tubes, and in each plot. And then every couple of days or so, he'd come in with a camera, a big round cylindrical camera that rotates down these tubes. It takes about 15 minutes, and it goes down taking pictures all the way down and then all the way back up. So it'll take pictures of the roots in the annuals, in the perennials, in the wild species, the intermediates. And I want to show you now some of the results in picture form. Okay, so here on the left, you can see the wild perennial wheat relative. So that's the That's the wild looking one that is perennial, that has low yield, that I showed you a picture of. And then on the right is an annual wheat variety that is grown by farmers typically now. And then in the center is our perennial wheat hybrids. And the numbers that you see, 0, 1, and there'll be others, that those represent the depth in feet of the soil. So I'll just show you what this looks like, and keep an eye on the roots as this goes through of each of these. So I think we'll look at it again, but hopefully you'll notice that the perennial wheat hybrid, the root system is really much more robust from the top to the bottom than either the wild perennial wheat relative, which it typically does have very good roots, and annuals as well. So we can see that these roots are critical and really important in both yield and perenniality here. Hopefully you all are on the seat of your pants, as am I, with excitement at this. And so how does this, how is this relevant to our research with health and nutrition? And so this is just one of the studies we've done. We've done others, or we have other results as well, but just looking at iron, manganese, and zinc, and comparing our perennial lines with annual lines, it's grown in the same field, same year, same location, everything. They are typically, the perennial lines have typically higher iron content, zinc content, manganese content, contents of other micronutrients as well. So this is something that the roots really are important in helping capture, as well as really helping prevent erosion, which is very important issue in our area of the world. And then it does also really kind of highlight the importance of soil in all of this, and how soil can play a role in different nutritional and health related traits. So we have these different soil scientists at WSU and cropping systems agronomists working with us on different questions, and some of these questions include looking at the beta-glucan content in barley, iron and zinc content in wheat, essential amino acid content in quinoa, metabolites and phenolic compounds in peas and lentils, and we're hoping that the list will keep growing here, as well as new graduate students are getting interested. And here's some of the graduate students working on this, and again, there are several others not pictured here as well. Okay, so the final example I'd like to give is on barley. And this is very exciting because I was a barley breeder here for 8 years, and I've recently handed this off so I could focus more on breeding for nutrition. And we released 2 varieties of barley. One is called Megsong, and the other one is called Havener. And then this is also where I'll give the background and kind of the foundation of our work in barley, and Julianne will then take this and kind of bring it more into the realm of the College of Medicine and the work that she's doing there. So very exciting. Okay, so here is a barley breeding field. We have several of these every year. What you're looking at is, this is on Spillman Agronomy Farm near Pullman, and each one of these fields has close to 1,000 plots. And as you look through here, you should be able to notice, so these plots are 20 feet long by 4 feet wide. So this picture that you're looking at is a typical breeding field with this many plots. So we often have thousands and thousands of these every year in this size or smaller size plots. And we have to go from thousands down to really to 1 or 2 that will be released as a variety. And it often takes 10 years from the time you make a cross in the greenhouse to the time you can release a new variety. And during that time, you select for yield, you select for disease resistance, you select for quality traits, functionality traits. Very, very, very rarely is nutrition involved in this selection process. And so that's what my lab is trying to change through our high throughput nutritional phenotyping lab. So we can work with the barley breeder now, we can work with the wheat breeders now, and test thousands of their lines every year for nutritional value as well. And so, but again, just using barley as an example, keep this field in mind. This is almost 1,000 lines, and we test each of these every year for beta-glucan content. And Julianne will talk more about beta-glucan, but I will just say that at this point, it's a heart healthy compound found in oats and barley. So here are the 1,000 lines, and you can see they range from 2% to about 9.5% beta-glucan, with most of them falling in about the 4% range. Those in the red circle are what we call high beta-glucan varieties. But essentially these are breeding lines, so they're not varieties yet. And the reason we don't just take some from the right, from that red circle, and release them as varieties, any of them, is yield. Yield typically decreases as beta-glucan increases. So our role, our job as barley breeders, plant breeders, is to look for exceptions to the rule. So over the past 3 years, we've found some of these exceptions and released them as new varieties. So Havener was our first new variety, and that had about 6% beta-glucan. Meg Song was our second variety, and that has about 7.5% beta-glucan. So we have varieties now with 50 to 70% higher beta-glucan than traditional varieties that you may have eaten. And I'd love to ask you how many of you have eaten barley in your lives or recently. Raise your hands. But if you have, odds are that barley is either a malt barley that failed to make malt quality that year, in which case it would have very low beta-glucan content, or it's a feed barley, so it was developed and bred for animals. And so that also has not been selected for beta-glucan content. So it might have 3.5%, 4%. Both of these are still delicious. They're both good for you. But our question is, are they actually as good as they can be? Can we make them better? Will improving beta-glucan content have an impact as well? So that's the idea between those new varieties, but it's not just varieties. So again, we have to test how each of these varieties, so here that means genotype, perform across different environments. So we've tested 20 genotypes in 8 different environments over 2 years. This was work done by Hallie Choi, all in the Palouse, and looked at how that impacted beta-glucan content, and are able to now identify varieties that have stable high beta-glucan across environments, because we want, if a farmer grows high beta-glucan content in one area of Washington, we want it also to be high in another area. So this is really important research. And then the next question is, every farmer grows their barley differently. So is there a way we can kind of figure out how to grow barley to optimize beta-glucan content? And this was work done by Cedric Javier when he was a PhD student in my lab. Now he's a postdoc in my lab, and he's looking at the effect of different nitrogen rates and seeding rates and other things to see how those impact beta-glucan content on barley. And then, so moving through this pipeline, once we have these new barleys, we also have to figure out how they perform, or even before they're new barleys. We send them out to the School of Food Science, to different food scientists around the country, actually, and they do different functionality tests. So these are loaves of bread, hearth bread that have been baked with 3 of these varieties. One of them on the left buck is from Oregon State University, and you can see they all make beautiful loaves of bread. These are 50% barley, 50% wheat, and this offers a really nice opportunity to incorporate some of these new high beta-glucan barley varieties into very commonly eaten products such as wheat. And then here you can see on the lower picture that the color of these varieties is quite different from each other, and there's all kinds of exciting things to do there as well. So one of the varieties, Meg Song. I want to introduce you to Meg. This is actually Meg Galnick. This is her variety growing in the background in Othello. And Meg, I'm bringing this up because Meg is a volunteer coordinator at the Pullman School Districts. And so she, along with a couple of my grad students, have been working to get barley on these high beta-glucan barleys, especially Meg Song, which is named after Meg, onto the menu. And so a pilot study's been done. The kids loved it as in the pizza crust. And so by next year, our plan is to get Meg Song grown for the school and into the school menu now. And again, this would just be a start, but really, really exciting stuff. Another thing we do is we work with a chef network that we've worked with chefs on these barley varieties from across the country and in different parts of the globe as well. Really, really great influential chefs. And what they do, we send them like our top 10 or 20 barley varieties, and they test them for different flavors and functionalities and how they would work much better than we can do in our lab. This is what they do. And then they advise us on which barleys they would be interested in growing. And as a result of this, Meg Song has been contracted out by a major food corporation and will be made available hopefully by next year. So it's a really new variety. It takes a little time, but hopefully by next year it will be out there for consumption. And Meg's barley has been highlighted by Bon Appetit. This is Daniela Soto Ines. She's one of the best chefs in the country, most well known as well. She has a restaurant in New York, and this is her Meg Song barley dish here. Okay, so I just want to back up and kind of go through again the soil to society pipeline that I did earlier, but this time with barley. As I finish up the talk here. And you can see the question here, which I'll get to in a second. But so again, starting with all of our work, breeding and agronomy and high throughput nutritional phenotyping in our lab, a lot of field work, often many years involved in this, working with soil scientists, working with cropping system scientists to help figure out how to grow these to optimize beta-glucan content. And then working with food scientists to develop new food products for this barley as well as this moves through the pipeline. And then this is where we get to ask this very exciting question. What are the cardiovascular impacts, if any, of eating varieties of barley with different beta-glucan content? So what this can provide a plant breeder such as myself is a target value to shoot for, and that's really what we need. I need to know if 4% is really sufficient for improving cardiovascular impacts from eating barley, or do we need 6% beta-glucan, or 8, or 10, or 12, or 14% beta-glucan? There's all these kind of questions we need to get at, and once we have that, then we can really hone in and focus on that as breeders and agronomists. And so again, this is work that is happening now, funded by WSU BioAg, by the Washington Grain Commission, WSU Strategic Reallocation Initiative. So again, here's Julianne Kellogg, and she's my PhD student in crop science, but she has a major project in her PhD to ask this question. And so she'll be talking about her research here with Petey and Pablo in the College of Medicine. And so at this point, I'd like to finish my talk and again invite you to email me, kmurphy2@wsu.edu, if you have any questions or just want to discuss further. I would love to be in touch with you all. And now turn it over to Julianne, where she can really focus on this last critical part of this barley and breeding pipeline for health and nutrition. So again, thank you very much for listening. Thank you, Celestina, for the very kind invitation. And I look forward to meeting you all face to face sometime in the future. Okay, take care. Thank you.